Good morning, students. Now we are going to discuss about the sexual reproduction. The sexual reproduction means the union of gametes, male and female gametes, that is called the sexual reproduction. The formation of the sexual reproduction is called gametogenesis. Gametogenesis. Okay. Next, the flower. The flower is three main stages. It possesses the changes included the three stages. They are pre-fertilization, fertilization and post-fertilization changes. The flowers is three stages. One is pre-fertilization, fertilization, third one post-fertilization changes. Okay, first we are going to discuss about the pre-fertilization changes. Pre-fertilization changes. Okay, in the pre-fertilization changes, it is hormonal and structural changes. Hormonal and structural changes involved plants lead to differentiation and development of floral primordium. Floral primordium, that is the young stages of the flower. The young stages of flower. That is the pre-fertilization changes. So one is the hormonal and structural changes is occur. It will leads to differentiation and floral primordium, development of floral primordium. Okay, next one is the male reproductive part. Male reproductive part, that is an important. The male reproductive part is called the Androecium. Male reproductive part is called the Androecium. The flower shows the four main parts: Androecium, gynoecium, calyx and corolla. The essential part, one of the essential part is called the Androecium. Now we are going to topic is the Androecium, the male reproductive part. See, the Androecium consists of a group of stamen. The individual stamen contains filament and anther. Individual stamen contains filament and anther. The anther contains microspore or pollen grains. Microspore or pollen grains. How to develop the anther? That is the development of anther. It is a four-lobed structure. The anther is a four-lobed structure. Each lobe contains a single row or a few rows of hypodermal cells become enlarged. Hypodermal cells, it will become an enlarged stage, that is called the anther. So the single archisporial cell, see, listen here, the single archisporial cell divides periclinal, that is a periclinal division. The periclinal division. So the single archisporial divided into two archisporial cells. One is called the primary parietal cell towards the epidermal layer. The second one is the primary sporogenous cell. It will lead to microspores and connective etc. So see, the primary parietal cell towards the epidermis, it will divide anticlinal and periclinal. See, the anticlinal, listen here, single, that is the archisporial cell, divides into two. Again, the archisporial cell, listen here, the primary parietal cell towards the epidermis and divide anticlinal and periclinal. See, anticlinal division, then periclinal division, anticlinal and periclinal division. So repeatedly to form 2 to 5 layer of anther wall layers. See, 1, 2, 3, 4. So 2 to 5 layer of anther wall layer. This is called the single sporogenous, primary sporogenous cell. The parietal cell divides the 5 layers. See, the anther wall, endothecium and middle layer and tapetum. So, epidermis, endothecium and middle layer and tapetum. Okay. Next one, the wall layers. Mature anther having the structures. See, the anther wall, endothecium, middle layer and tapetum. Okay, listen. The first one, epidermis. Epidermis is a single layered structure, protective, anticlinal and divisions to have the internal organ, to protect the internal organ. See, the epidermis is a single layer. It is divided in anticlinal division. Then it will protect internal layers. That is called the epidermis. The second one is endothecium. It is Generally single layered. It is a single layer. Second one is called the endothecium. It is a single layered. See, listen here. It is radially elongated and found below the epidermis. Below to the, it is a single layer, radially elongated below to the epidermis. That is called endothecium. Okay, the endothecium, it is a single layer wall layer, possess alpha cellulose, sometimes lignified. Underline the word alpha cellulose and lignified. Lignified means made up of lignin. It is a hygroscopic in nature. It is water living plants. Hygroscopic nature. That is an aquatic plants. So the next one, the anther lobe is two-lobed, that is a bilobed structure, that is four-lobed. And stomium means that is a single lobe. If the anther is a single lobe, it is a dehiscing property. Anther is a dehiscing property. Dehis the anther at maturity stage. At open the anther wall layer, inside the pollen grains are liberated. Outside, that is a stomium. Okay, next one, middle layer. The middle layer is also 3 to 4 or 5 layers repeatedly anticlinal and periclinal division to form the middle layer. Okay, the fourth one is tapetum. The tapetum is a very, very important one. The tapetum is innermost wall layer of the anther. The Androecium development. Okay, listen, the tapetum is a two type. One is secretory tapetum or parietal tapetum. Okay, next one, invasive tapetum. Secretory tapetum. The secretory tapetum contains the parietal cellular and glandular. The other name of the secretory tapetum, parietal, glandular and cellular. The invasive tapetum, the cells possess the lose their wall layer. The individual wall layer of tapetum, listen, tapetum cells lose their wall layers. The cells contain protoplast, nucleus, everything. The wall layers are disintegrated. Listen, the wall layers are disintegrated. It contains only in the plasmodium, that is a protoplast. All the protoplasts are coalesces to form periplasmodium. Okay, the secretory tapetum retain there its original position. Like this. Original position. It gives the nourishment to the microspore. That is a tapetum wall layer. Okay, secretory tapetum and invasive tapetum. Okay, next one, anther lobe or anther cavity. See, the anther cavity, it is a microspore. Anther cavity which contain the microspore. The microspores is in young stage. At maturity, it is a pollen grain. At maturity, it is a pollen, called as the pollen grain. The next one is connective layer. The connective, this is sterile tissue surrounded by the anther lobe. Total anther lobe contain a connective layers. It possesses vascular tissues. The xylem and phloem of the vascular tissues are originated in the connective tissues. It contributes to the inner tapetum. So, the wall layer of the tapetum, it will give the nourishment to the inner wall layers. Okay, that's all about the topic. Okay, students. Next one is microspore or pollen grains. The microspore have protoplast surrounded by a wall layer. Microspore contain the protoplast. See the microspore surrounded by the protoplast having the wall layers. The pollen protoplast consists of dense cytoplasm with nucleus. The cytoplasm with nucleus. So, dense cytoplasm with the nucleus. There are two wall layers. One is intine and another one is an exine. The intine wall layer is very thin layer. Then this is an uniform and made up of cellulose, hemicellulose, pectin, callose with protein. The intine wall layers say The wall layer is outer layer is called exine and the inner wall layer is intine. The inner wall intine is a thin walled layer, uniform, that is all layers are jointed and made up of cellulose, hemicellulose, pectin and callose with protein. That is the intine. The next one is an exine. Exine is thick wall layer. Exine is a thick wall layer. It is interrupted, made up of cellulose, sporopollenin and pollen kit. Sporopollenin and pollen kit. That is an exine layer, exine wall layers. See, the sporopollenin means the exine layer is made up of sporopollenin. It is absent in germ pore. Absent in germ. There is a germ pore. Germ pore area there is no sporopollenin. Sporopollenin is absent in germ pore area. Surface of the exine is smooth or various pattern. That in shape is a different one. The shape of the pollen grain is varies from species to species. What are they? Globose, ellipsoid, and fusiform. Globular, annular. That is the shape of the pollen grain. The shape of the pollen grain is varies from species to species. What are the shape? Globose, ellipsoid, fusiform, lobed and angular. The next one is the pollen kit. These are the 3 more questions: sporopollenin and pollen kit. The pollen kit contributed to tapetum, colored. It will produce a colored yellow or orange color in pollen kit. It is made up of carotenoid and flavonoids. It attract the insects. The colored pollen kit, it attract the insects and protect the damage from ultraviolet radiation. That is a pollen kit. So pollen kit and sporopollenin are present only in the exine area. Exine area is interrupted. The germ pore areas. The germ pore areas are not absent in germ pores. Okay. The next one is development of male gametophyte. See, the microspore are the first cell of the male gametophyte. Microspore is the first cell of the male gametophyte. It is an haploid in nature. The development of male gametophyte take place while they are similar in microsporangium. Nucleus of the microspore further divided into, see, the nucleus of the microspore further divided into nucleus of the microspore divided into 2. The nucleus of the microspore divided into 2 still in the microsporangium. The microspore divided metatically to form vegetative and generative nucleus. See, the nucleus is divided into generative and vegetative nucleus. Listen, next one, the vegetative nucleus and otherwise called the tube nucleus. The generative nucleus again divided into 2. Generative nucleus another divided into 2. That is male gamete. We called as the microspores. Listen, here the nucleus is divided into 2, that is generative nucleus, otherwise called the generative nucleus. The generative nucleus divided into 2, that is 2 male gametes. The vegetative nucleus otherwise called the tube nucleus. Here the outer layer is called the exine. Exine is thick walled cells. The thin walled pollen inner wall layer is protrude outgrowth to form a pollen tube. That is a pollen tube. The pollen tube grows towards the outside. The male gamete and tube nucleus are carried together and stigma. It will enter into the stigma. To form fertilization process. That's all about the microspores or pollen grain. Thank you, students.